Good morning, good afternoon, good evening, everyone. Welcome to the last presentation of Session 5, The Global Nature of the Generic Industry. My name is Wenlei Jiang. I'm a Senior Advisor for Innovation and Strategic Outreach at the Office of Research and Standards, Office of Generic Drugs, CDER, US FDA. Today, I'm going to present how can scientific advancements help align global development of complex generic products. This presentation reflects my own view and should not be construed to represent FDA's views or policies. The concept of complex product was introduced in GDUFA II commitment letter. FDA provided a list of complex products, including products containing complex active ingredients, complex formulation dosage form, complex route of delivery, complex drug-device combination. Here, I want to emphasize, in the context of GDUFA II, complex products are the ones which present challenges for generic drug development. That is, difficult to assess drug substance sameness and/or bioequivalence at the intended site of action. The nature of product complexity is diverse. Some product complexity is associated with drug substance, some are with dosage forms. Also, different regulatory agencies may have different classification about complex products. The concept of complex product is not static. It may evolve over time based on industry feedback and agency experiences. Based on published guidelines and approval summaries from different regulatory agencies, we know that different regulatory standards have been used for the approval of important complex generics or follow-on products in different jurisdictions. Some products may have been approved by EMA, Health Canada, but not by FDA, or vice versa. Harmonization of technical and scientific standards for generic drugs presents an opportunity for significant public health benefits by streamlining drug development across regulatory jurisdictions and increasing patient access globally to high-quality, affordable pharmaceuticals. Efforts were initiated with the ICH bioequivalence guideline development of immediate release solid oral dosage forms. As stated in the ICH reflection paper on generics endorsed by ICH Assembly in November 2019, a series of ICH guidelines on standards for demonstrating equivalence of more complex dosage forms will also be followed. In the next 10 minutes or so, I will highlight US FDA efforts, especially GDUFA research and global collaboration efforts to overcome scientific and regulatory challenges to help development of safe, effective, and high-quality complex generic drugs. First, a good news to share. About 3 weeks ago, FDA published the Manual of Policies and Procedures, abbreviated as MAP, on classification of complex products. In this map, definition and examples of complex drugs and drug-device combination products were provided. In addition, the responsibility and the procedures for complex drug classification and the database maintenance are also described in the map. This map helps clarify understanding about complex products and the standardized complex product classification process, making complex product classification transparent to industry and any regulatory agency who may be interested in understanding FDA's complex product concept. This slide summarized some analytics to highlight US FDA regulatory science efforts to support generic drug development and approval between fiscal year 2018 and 2020 in the following 3 categories: development of generic drug products, generation of evidence needed to support efficient review and timely approval of ANDAs, and evaluation of generic drug equivalence. As shown in the table on the right corner, highlighted in green, the number of ANDA approvals impacted by research has a steady increase from 63 in fiscal year 2018 to 152 in fiscal year 2020. These are solid data showcasing scientific advancements helped development of generic products. If you are interested in more of these analytics, please refer to this web link for more details. Next, I will select 3 complex product categories to illustrate the research focus in each product category and the impact of GDUFA research on guidance development and product approval. The first is about orally inhaled and nasal drug products, abbreviated as OINDPs. Here is a cartoon to summarize B requirements for OINDPs from different regulatory agencies. The left end is weight of evidence approach, and the right end is stepwise approach. US FDA and EMA are at the two opposite ends. PMDA and NMPA is closer to US FDA's weight of evidence approach. TGA is more aligned with EMA's stepwise approach. In recent years, Health Canada and Anvisa are shifting slightly more towards the stepwise approach. So what's the weight of evidence approach for OINDPs? That is, the generic applicant need to demonstrate formulation sameness and device similarity between the generic and the reference product. On the basis of formulation sameness and device similarity, the generic product and the reference product should demonstrate equivalence in product performance, equivalence in systemic exposure by plasma PK studies, and equivalence in local drug delivery by comparative clinical endpoint or pharmacodynamic BE studies. Not each individual equivalence metric, but all should be met in capturing all aspects of an OINDP's performance. In contrast to US FDA's weight of evidence approach, EMA adopts a stepwise approach to establish bioequivalence for OINDPs as illustrated in this slide. Step 1. Conduct in vitro equivalence tests. If equivalent, TE is concluded. If not, move to Step 2. Conduct PK studies with and without charcoal blockage to demonstrate equivalent pulmonary deposition and total systemic exposure. If equivalent, TE is concluded. If not, move to Step 3. Conduct pharmacodynamic clinical studies to demonstrate local equivalence. As you can see, there is significant difference between weight of evidence approach and stepwise approach, presenting additional difficulty for generic applicants who intend to file OINDP applications in multiple jurisdictions. Recently, FDA had alternative thinking regarding CCEPs or PD studies for OINDPs. CCEP and PDB studies are considered less sensitive for evaluating formulation differences than other B methods due to high variability and flat exposure response of these studies. FDA encourages applicants to propose alternative approaches to the CCEP and PDB studies. Hopefully, these alternative approaches can address the relationship of systemic PK to local drug levels within the lungs, address the relationship between in vitro performance to local drug deposition and clinical performance. These alternative approaches include, but are not limited to, modeling and simulation and advanced analytical methods for better characterization of the product. Here, I would like to give you an example of advanced analytical method for better characterization of the product. Morphology-directed Raman spectroscopy, MDRS, combines microscopic imaging and Raman spectroscopy in one integrated platform for substance-specific particle size determination. This method helped determine the particle sizes of the API, excluding interference from other excipients. This technique has supported the approval of first generics of mometasone nasal suspension. We will continue to explore this method's potential to reduce some equivalence tests for other OINDPs. Some research funded by FDA demonstrated that realistic in vitro aerodynamic particle size distribution methods incorporating mouth-throat models and a realistic range of inhalation profiles can provide a better prediction of deposition of inhaled particles in the lungs and capture patient variability. Also, in vitro dissolution system such as transwell system was optimized to characterize release profile of fluticasone dry powder inhaler and metered dose inhalers. This method can adequately capture differences in formulations. The advancements in vitro studies provide options for alternative BE approaches to CCEP, helps promote regulatory convergence and improve global access of OINDPs. In topical dermatological product area, FDA research has focused on supporting the expansion of characterization-based BE approaches to a majority of topical dermatological products, developing PK methods to directly monitor the drug absorption at or near sites of action in the skin, and enhancing PBPK models to predict the drug absorption. Here are some example draft product-specific guidances related to topical dermatological products based on GDUFA research outcomes. Also, in recent years, there are a significant number of generic topical dermatological product approval, which will help improve patient access to these products at a lower drug price. As of long-acting injectable drug products, there are quite some challenges as well as opportunities ahead of us. Currently, no generic version of long-acting PLG microsphere product is approved by US FDA. GDUFA research has focused on developing new tools for complex excipient characterization, understanding how raw materials impact formulation characteristics and drug release, and exploring more clinically relevant in vitro release methods, and others. These research outcomes were translated to support product-specific guidance development and generic drug approval. To help align global development of complex generic drugs, FDA has been utilizing some established platforms, including ICH Generic Drug Discussion Group and International Pharmaceutical Regulators Program, IPRP, to have direct dialogues with global regulators. In addition, some scientific topics on bioequivalence requirements were discussed at the Global Bioequivalence Harmonization Initiative workshops among regulators, academia, and industry scientists. In 2021, FDA launched 2 new initiatives, including Generic Drug Cluster and FDA EMA Parallel Scientific Advice Pilot Program. The Generic Drug Cluster is the first forum established for the world's leading regulatory agencies to address generic drug development globally, aiming to increase scientific alignment among leading generic drug regulatory agencies. As of FDA EMA Parallel Scientific Advice Pilot Program, the goal of this program is to provide a mechanism for FDA and EMA assessors to concurrently exchange their views on scientific issues with applicants during the development phase of complex generic drug hybrid products. Through the PSA process, applicants would gain an understanding of both agencies' recommendations. Next, I would share with you some discussed topics at generic drug cluster meetings, including parenteral nanomedicine, long-acting injectable products. These products presented significant scientific and regulatory challenges, for example, potential partial AUC considerations, physicochemical characterizations. Reaching convergence regarding approval standards of these products will help global patient access to these products. Our discussion focused on pinpointing key differences among different agencies regarding regulatory standards, identifying aspects hindering approval, and exchanging regulatory scientific advancements. Hopefully, fruitful exchange and discussion at the generic drug cluster and other global collaboration platforms can serve as basis for future ICH guideline development of complex products. In summary, FDA is committed to support complex generic product development through GDUFA research and GDUFA 2 enhancements. I hope now you can understand that though there are unique scientific and regulatory challenges for global development of complex generic products, significant regulatory science advancements have facilitated guidance development and approval of complex generic products. Furthermore, global collaborations help accelerate scientific advancements and align global development of complex generic products. With that, I would like to thank my colleagues at Office of Generic Drugs, Office of Pharmaceutical Quality, GDUFA research collaborators for their contributions to the work presented here. Now, I will pass the podium to Sarah.